You might have seen this guy before. If not, well, it's a DNA. But what is the DNA? The DNA can be found in the cell's nucleus, well, at least for eukaryotic cells. Our DNA carries our genetic material. It is made up of a double helix twisted together like a ladder. Each of the strands contains sugars and phosphate groups. These are called nucleotides. There's adenine, thymine, cytosine, and guanine. These bases are connected by hydrogen bonds. In the DNA, thymine binds with adenine and guanine binds with cytosine. Now, imagine that every cell is a tiny, self-powered factory that runs on a genetic instruction manual that are made up of letters. For a long time, we could only read these letters in the manual. But with synthetic biology, scientists are now able to read the manual plus revise it. We can now treat DNA as an editable code and redesign cells to make something new. You may or may not have heard of CRISPR before. CRISPR stands for clustered, regularly, interspaced, short, palindromic, repeat. But what is CRISPR? CRISPR is a form of immune response in bacteria to protect themselves from viruses like bacteriophage. Think of your body as a 3 billion letter recipe book. If one word is misspelled, the recipe can fail and cause a disease. What happens then? Using CRISPR, scientists can look for this misspell and fix it. It can work as a search, cut, and replace command. But how does CRISPR do that? The CRISPR components include the target DNA, Cas9 enzyme, guide RNA or gRNA, and the PAM sequence. Once a bacteria detects the presence of a pathogen, it produces a gRNA containing the complementary sequence of the virus. The Cas9 is a nuclease enzyme that is able to act as a molecular scissors cutting DNA. However, it can't function on its own. It needs the help of the guide RNA. Together, they form a complex to look for the target sequence. Once the guide RNA finds the target sequence, the DNA unwinds, and the gRNA binds to the DNA via complementary base pairing. The Cas9 then cuts the DNA, which ends up killing the virus. But how does the Cas9 know where to cut? This is where the PAM sequence plays an important role. The PAM sequence, or also known as the protospacer adjust motif, helps the Cas9 identify between self and non-self. It is usually located 2 to 5 base pairs downstream of the target DNA sequence. The PAM sequence is different for each organism and also depends on what Cas enzyme is used. For example, in Streptococcus pyogenes, the SP-Cas9 recognizes NGG and being any base, while in Streptococcus canis, the SC-Cas9 recognizes NNG. CRISPR works like a pair of tiny, programmable scissors, giving researchers precision, speed, and universality. Because every organism, from bacteria to plants to people, carries DNA, CRISPR can be adapted to edit nearly any living thing. In fact, in 2020, Jennifer Doudna and Emmanuel Charpentier won the Nobel Prize for Chemistry for their groundbreaking work on CRISPR-Cas9 gene editing technology. They demonstrated that by designing a short guide RNA complementary to any target DNA sequence, the CRISPR-Cas9 system can be programmed to make precise cuts, allowing scientists to remove, insert, or replace genetic material at exactly the site you choose. However, it is also important to note that CRISPR creates double-stranded breaks that are prone to errors like indel mutations. With this, Scientists wanted to figure out a way to edit bases without the need for double-stranded breaks. This is where base editing comes in. You might have heard the news of the world's first personalized CRISPR therapy that is given to a baby born with CPS1 deficiency. It is a disease that has an estimate of 50% mortality in early infancy. Using a corrective CRISPR-Cas9 technology, or base editing, scientists were able to give personalized treatment to this baby to correct his specific disease-causing mutation. While CRISPR acts like a scissor and glue, base editors work like a pencil and eraser. There are two types of base editors. There are cytosine base editors and adenine base editors. In a cytosine base editor, cytosine is converted into uridine via deamination. During DNA replication, uridine is read as thymine. In an adenine base editor, adenine is converted into inosine by adenosine deaminase, which is then read as guanine during replication. So, how is base editing a great tool? Base editing offers powerful, precision, find and replace approach to fix single-letter genetic typos without chopping the DNA backbone. By harnessing the cell's own repair machinery, cytosine and adenine base editors can swap out disease-causing bases efficiently and with minimal off-target effects. As delivery methods and editor specificity continue to improve, base editing holds immense promise for both fundamental research and personalized therapies targeting point mutation disorders. These advances aren't just laboratory curiosities, they're already saving lives. The world's first personalized CRISPR therapy for the baby with CPS1 deficiency shows how we can correct disease-causing typos in a patient's own genome. As delivery methods improve and editor accuracy continues to climb, CRISPR and base editing will touch everything from medicine to agriculture to fundamental research. Yet, with great power comes great responsibility. As we learn to write and rewrite the language of life, we must navigate ethical considerations, regulatory frameworks, and safety concerns with equal care. If we get it right, synthetic biology will open doors to treating genetic diseases once deemed untouchable, engineering sustainable solutions for our planet, and answering some of biology's oldest questions, one letter at a time.